Guys, this is the last video in our protein synthesis series, and this one we're actually going to talk about how the proteins get made. What you see here is a growing protein. This is a little fuzzy, but we can see that we're making a protein. So let's refresh our memories as to what translation is. Translation is the process by which nucleic acids are turned into, or the codon nucleic acid create proteins. And if you remember our nucleic acids, we start off with DNA and then we go to mRNA. And what we're going to see today is then we go to tRNA. And then we finally get to our protein. So what we're going to do is we're going to kind of break this down step by step. Let's talk first of all about what we're seeing on this page. So in gray, we have a ribosome, and there's two parts to a ribosome. They're called subunits, which you don't need to know, but if you're interested, the larger subunit and the smaller subunit. Now, when the mRNA, which is here, so this strand is mRNA, it's at the nucleus. These ribosome pieces aren't together. And what happens is the two ribosome pieces snap together like an Easter egg. If you ever did an Easter egg hunt when you were a kid and you had the plastic eggs and the top side's larger than the bottom side and they snap together, that's how it connects. And then the mRNA gets sandwiched in between. On the ribosome, we have three sites where the tRNA combines. We have site 1, site 2, and site 3. And that's the first thing that's going to happen is the ribosome units in the mRNA are going to come together in a complex. So number one, the mRNA plus the ribosome assemble into a complex. Assemble into a complex. What will happen is the ribosome, you'll see down here, it says, it's a little fuzzy, but it says movement of ribosome. The ribosome actually moves along the mRNA strand. And once it hooks together, it'll continue, it'll just kind of begin to slide nucleotide by nucleotide down this mRNA strand. And we won't start protein synthesis until we get to a specific codon, and I'm going to pause. I need to go grab my codon sheet. Okay, I finally found a sheet. It was one that I'd already written on, but it'll work. So you noticed earlier when we used the mRNA to the amino acid chart that these are the mRNA codons, that we had 3 stop codons. So if we have UAA or UAG or UGA, that will cause translation to stop and the protein will get released. But there isn't one that says start, but you notice down here it says MET. The codon for MET is AUG. This is your start codon. So the first amino acid in every protein is methionine, MET, because this is what signals the start of translation. So let's go back. And the second thing that's going to happen, I think I got my paper all wonky. The second thing that's going to happen is when the ribosome encounters AUG, translation will start. Again, we can have a lot of nucleotides before it, like maybe we're going to, like we're going AAA, TTT, GCC, GCA, but when we hit this AUG, that's when things get hopping. Now we talked about these sites, and on this diagram, this represents, so this is the amino acid, this is the tRNA, the body of the tRNA, and this is my anticodon. So I have a U, a C, and a G here. This is another tRNA molecule, and its amino acid is here, and this has A. And then this is a tRNA that is leaving. It has the anticodon CCC, and you'll notice it has no amino acid. And the reason for this is that it has attached its amino acid to the protein chain. So how does this work? Once we hit that AUG, the tRNA or a tRNA molecule with the anticodon. So before I write it out, what would the anticodon be? If I have AUG, what is the tRNA molecule going to have on this side? Did you say UAC? If you did, you're right. So the tRNA, the first tRNA molecule that came in, if we draw it the way they drew it, had UAC on one side, and it has methionine on the other. So that molecule came in and it attached to site 1. So it attached to site number 1. And then the ribosome is going to move another set of codons. So the ribosome moves. And this is going to shift that tRNA molecule to position 2. Call these sites. Shifts it to position 2. It's at position 2 that the amino acid on the tRNA gets attached to the growing chain of the protein. Now, obviously, for this first one, there was nothing attached to because it was the first amino acid. But usually what's going to happen then, I'm going to start up here, number 5. At site 2, the amino acid gets added to the chain. We call it a polypeptide. That means that this first amino acid up here had to be methionine. So as this moves, the next tRNA comes in and binds to site 1. And then we're going to shift again. So the ribosome is going to move again. And what we're going to have is at this first site, so it's site 1, switch colors here. At site 1, a new tRNA is going to come in. At site 2, the amino acid, and I'm going to abbreviate that AA, binds to the protein chain. And at site 3, the tRNA minus its amino acid, so it doesn't have an amino acid anymore, leaves. And we'll see this is going to happen again and again and again and again. And we'll just keep adding amino acids until, what do we see for that last one? So this is going to continue until we reach what? Until we get to a stop codon. When we get to the stop codon, this really, and this can be hundreds of thousands of amino acids long, gets released. And right now it's just a single straight line. You remember we talked about the fact that it has to go through a folding process. So we call it, when it's just a single straight line, we call it its primary structure. Then it's going to fold into like an accordion folded sheet or a curlicue ribbon. That's secondary structure. The tertiary structure, we start to stick some of those different shapes together. And then finally, at the end, we end up maybe attaching some inorganic things or, you know, putting some extra molecules on, kind of like how you put earrings or jewelry on an outfit, if you're a lady. So again, what are our takeaways? So this is the process of translation. We're finally going here from our nucleotides to here as a protein, and it happens with the tRNA. We get this complex that assembles. The ribosome moves along the mRNA till we get to a start codon. At that point, the tRNA with the right anticodon and methionine comes in, because that's always how our proteins start. And then we begin this process of moving everything to down. The ribosome moves down the mRNA, and we keep adding a tRNA to the site 1. The amino acid binds at site 2, and then the tRNA you see here is released at site 3. And this continues over and over and over. By the way, what happens to this tRNA molecule? It'll go float around until it runs into another, let's see, if this is CCC, the anticodon for that would be GGG. And when I look, I see that's glycine. So this was a glycine. So this will continue or roam around until it finds a glycine, and it'll reattach. Okay, that takes care of all the high points of translation. We're going to do a lab over this and do some review. Thanks for watching, and that finishes off this chapter.